Right, okay, this unit here, or this key area in Unit 1 is about genetic engineering. And you hear, or maybe you've heard about genetic engineering or genetically modified crops for in the news or some of the debate around it, or you've heard about genetic engineering where you've seen little green pigs with wee green snouts or green cats, or you've heard something about spider thread being put into goat milk and so on. And there's a lot of debate over genetic engineering, whether it should be done and whether it shouldn't be done. But we're going to have a look at what genetic engineering is and have a look at a real example of it being used and being used for a very useful purpose. So to do that, we're going to have a look first of all at this disease here called diabetes. Diabetes is a condition where people aren't able to produce this hormone here called insulin. Now, insulin is involved in controlling the levels of glucose that are in your blood. And what the insulin does is make sure that the levels don't get too high. It keeps the glucose levels in balance. And we have a look at this in a wee bit more detail when we come to do the second unit. But basically, you just need to know that if the level of glucose isn't controlled, then ultimately it can cause a disease called diabetes, and a person can die from that. And it used to be in the past when people were diagnosed with diabetes, there was nothing that could be done. But then it was discovered that insulin could be injected into people, and the insulin came from cows and from pigs. Sometimes as well, it's come from horses too. Insulin comes from the organ, the pancreas. It's produced there in the pancreas. And so in order to produce it, you had to kill a cow or kill a pig. Now, obviously, there's some disadvantages to that. One of the disadvantages being that you're having to kill animals to produce the insulin. But there were other disadvantages with it as well. That was that some people just didn't like the idea of injecting something that came from animals, or it was also a pretty expensive process. And more importantly as well was that some people were actually allergic to animal insulin. And I've got an example of that. My mother herself is diabetic, and in the past she was using an insulin that was causing an allergic reaction and it had to be changed. And nowadays we can actually produce human insulin using genetic engineering. So, what do we mean when we're talking about genetic engineering? Well, what we mean by it is the transferring of genetic material from one organism to another organism. And what we're talking here in the context that we're speaking about is looking at moving the gene that produces insulin from human chromosomes and putting them into bacteria. So we're going to have a look at the steps in that process. Now, I've got an example here where I've spoken about insulin, but also this can be used maybe to produce as well human growth hormone as well. It's been used to do that too. So what we're doing is we're using a bacteria to produce a specific protein that we want, either insulin, human growth hormone, or something else. And the reason that we can use bacteria is because bacteria have got plasmids in them. And plasmids are rings of DNA found in bacterial cells, and they're easy to remove, to alter them, and to put them back into bacteria. So we're going to have a wee look at what goes on here. Now, I think I've got on the next slide here, just a little picture, yes, of our plasmids. So here we're looking at a bacterial cell, and if you remember back to bacterial cells, you remember they've got circular chromosomes, these large circular chromosomes. They don't have a nucleus. but they have these smaller circular pieces of DNA called plasmids, and it's these that we use as our wee genetic engineering tool. So looking at the process of what goes on here, we have a wee look at the diagram that I've got here. Over at this side, we've got part of a human chromosome. Remember, you've got, all these different 23 pairs of chromosomes in the body, 46 chromosomes in total. And on one of these chromosomes, there is a gene that will be involved in the production of insulin. And here it's just represented by this green colour here. Now what happens is the scientists, they can use a special enzyme to cut that gene out from the human chromosome. And as well as that, they can take a bacteria, like the bacteria that we had on the slide before, and remove the plasmid from the bacteria. So here's a plasmid here that's been removed from the bacterial cell, and that plasmid can be cut open. In actual fact, using the same enzyme that was used over here, it cuts open the plasmid, and then this gene can be taken and it can be inserted into where the cut has been made in the plasmid. And there's a special enzyme that is used to stick these or glue this gene into here or insert the gene. Then once that gene has been inserted into this plasmid, the plasmid can be taken and it can be inserted into another bacterial cell. And then from there, what happens is these bacteria can be grown, and what will happen is the scientists grow them in large fermenters, and they grow them with a whole lot of nutrient broth, so they give them lots of food and they keep them warm and they've got them just at the right temperature and they give them oxygen. So what happens is they start to multiply, so you get lots and lots of bacterial cells, and they've all got the little plasmids inside them. It's not a very good diagram, but they've all got inside them the gene that you want, because it'll have made copies of it, and that gene will go on to produce lots of useful substance, and in this case here we're talking about lots of insulin. And the bacteria are grown up. By grown up, what I mean is they're grown in these fermenter conditions, in these big vats, and they multiply and you produce lots and lots of this useful substance, insulin. And what happens is that is purified, and then the insulin is put into bottles, and then everything's kept sterile, of course, and then the insulin can then be used to be given out to people who are diabetic, and they take that insulin and they inject it in order to make sure that they've got insulin in their body whenever they eat and to keep their blood sugars level. Okay, so just as I said there before, the bacteria will multiply, the hormone then that you want can be extracted and purified, and the bacterium that has been changed, in other words, it's had this gene put into the plasmid, is known as being genetically modified. So it's a genetically modified organism, or a GM organism. And there are kind of obvious advantages to genetic engineering. One being, when we talked before, we spoke about the allergies. Now, if you're taking a human chromosome and you're taking a gene from a human chromosome and you're putting that into a bacterial cell, although it's a bacteria, it is actually producing human insulin because it's the gene for human insulin that is there. And so therefore, when that is being sold as an insulin product for people who are diabetic, then it means that it's human insulin that's being used, and so you're less likely to have an allergic reaction. Other advantages are, well, you can select some kind of single characteristic that you want. So if you're, say, genetically modifying a plant, for example, you could select a characteristic like maybe being resistant to some sort of herbicide, or resistant to some sort of disease. And you can produce the thing that you want, you can produce it very quickly. You can make sure that it's very pure, and I said also that it won't cause any allergies. And all of these things make genetic engineering a very advantageous process. Now, if I just move up here, we'll have a wee look at, I did mention before that there is a bit of controversy around genetic engineering and disadvantages of genetic engineering. Now, one of them is that I did mention that you've got genetically engineered bacteria, so you've put some kind of human gene into that genetically engineered bacteria, and sometimes there's a concern that that might end up in the environment and cause some kind of disease that might affect humans. Or it might be, and sometimes this is used when the bacteria is genetically modified. They actually separate out the genetically modified bacteria to, because what happens is when the bacteria are produced and the plasmid is in them, what happens is when you are trying to get that plasmid into the bacterial cells, some cells don't take up the plasmid, and they actually separate out the bacterial cells that have got the plasmid in it and the bacterial cells that haven't, by actually putting in a little gene for antibiotic resistance in there, as well as the gene that you want. So you only get the bacterial cells growing that have got the antibiotic resistance as well. And there's concern that the genetic engineering can transfer that antibiotic resistance to other bacteria that aren't being used in the genetic engineering process. Also, to develop this, it's pretty expensive as well, so that's another disadvantage that is there. But the thing is, once it's been developed, then people know how to do it, and so the costs do start to decrease. In the case of, say, genetically modified crops, there is a concern that if you've got a wee plant and you have given it some sort of advantage, say you decide that you want to make it resistant to some kind of disease, there's a concern that that disease resistant gene that is in this plant here, this is the one that you want, the plant that you want, that's okay, it ends up with disease resistant, but you might actually end up with some transfer in the pollen, ending up with the gene into something that you call a weed, and so you end up with a weed that you don't want, that's got some kind of advantage. And so that is a concern with genetically modified crops. And so sometimes there's quite a controversy over growing genetically modified crops. And there's special laws in this country over growing genetically engineered crops. Some countries do grow them, but in this country you need to have a special license and special permission to do that. And lastly, if we just have a look at this slide here. Oh, there we are. We're talking about DNA being transferred by us from, say, a human to a bacterial cell, but in actual fact, genetic engineering does happen in nature, because sometimes what happens is that bacteria themselves actually transfer bits of information, and they actually transfer plasmids from one bacteria to another. So they actually naturally transfer genetic information, or as well as that, viruses will actually transfer genetic information into either bacteria because there are bacteria that invade viruses, viruses that invade bacteria. As well as that, there are viruses that invade plants, and some of these viruses actually transfer genetic material into the plant cells. They're little natural genetic engineers. You don't need to know any more information about them, but you just need to be aware of the fact that DNA can be transferred in nature. So that's a summary of genetic engineering, and you can make some notes on this. Make sure you note in your notes the step-by-step process of genetic engineering, including from when the gene is cut out of the human chromosome and then the plasmid being cut open and so on. So you've got a wee flow diagram in your notes of the actual process.